Usually when we talk about a medical diagnosis, there's this underlying expectation of mechanical precision. Like if you break your arm, the X-ray shows that stark, jagged, white line.
Right against the black background. It gives
Yeah.
you a very definitive answer.
Exactly. The mechanics of the failure are visible, and, the protocol to fix it is completely linear.
And I think that provides a real sense of control. We inherently prefer pathology to be visible. We want it easily categorized into a binary state, like it's either functional or broken.
But then you step into the realm of neurodevelopment or trauma or neurodegenerative disease, and that X-ray machine is essentially useless.
Yeah, totally useless.
We are looking at a diagnostic landscape that is just incredibly murky. When dealing with cognitive decline diseases like Alzheimer's or Parkinson's, or even just that pervasive brain fog that so many of you listening accept as the inevitable tax of getting older, we're constantly hunting for that single jagged white line.
We want the simple fix. We search for the one broken mechanism so we can fix it with a highly targeted pharmaceutical intervention.
Right. A magic pill.
Which is, frankly, a fundamental miscalculation of how the brain operates. The reality of the human brain is an ecosystem of staggering complexity. The solution to its degradation isn't necessarily a targeted chemical hammer from the outside.
And the sources we are synthesizing today for this deep dive suggest something radically different.
They absolutely do.
The solution is often about activating a dormant, deeply encoded healing intelligence already hardwired into your biology. Modern medicine frequently treats cognitive decline as an inevitability of the passage of time, like, simple wear and tear.
But our bodies, specifically our neurological networks, possess an innate mechanism for profound cellular renewal. We are not naturally destined to decay at the rate we currently observe in modern society.
The problem is that our modern industrialized lifestyle has forcefully flipped the switch on that repair mechanism to the off position and basically locked it there.
We've jammed it shut.
Yeah. So the mission of this deep dive isn't to look at a list of symptoms. We are mapping out the biological magic trick that flips that switch back to cellular renewal. We are exploring the science of fasting.
And I think we need to be very clear here.
Oh, absolutely. To be absolutely clear with you right up front, this has zero to do with fasting to fit into a pair of jeans or tracking macros or shedding water weight. We are going to look at how specific metabolic states orchestrate a biological symphony to heal, protect, and physically rebuild the human brain.
But it does require an entirely different vocabulary. To understand how you can manipulate this metabolic switch, we have to navigate the mechanisms of, well, it's a mouthful: neurogenesis, macroautophagy, mitophagy, xenophagy, and agrophagy.
Okay, let's unpack this because they sound like dense terminology from a postgraduate pathology textbook, I know. But these are highly relatable, dynamic processes happening or failing to happen inside your head right now.
Exactly. They are the mechanics of survival.
So before we can even begin to understand how the absence of nutrients triggers the brain to heal itself, we have to establish why the brain is so incredibly vulnerable to damage in the first place. Why are our neurons uniquely fragile compared to, say, the cells in our liver or our skin?
Well, it comes down to three distinct biological vulnerabilities. The first is their lifespan. Neurons are characterized by long, post-mitotic lifetimes.
Post-mitotic, meaning they've exited the cell cycle, they're no longer dividing, right?
Precisely. If you look at an epithelial cell, like a skin cell or a cell lining your gut, they are constantly undergoing mitosis. They replicate, they divide, and as they do, any intracellular damage, misfolded proteins or metabolic waste gets inherently diluted.
It's like sweeping the dirt into a new room and then throwing away the old room.
That's a great way to look at it. Eventually, those older cells undergo apoptosis, programmed cell death, and simply slew off, taking their accumulated trash with them. It's a continuous, rapid cycle of renewal through division.
But neurons don't get to do that.
Neurons do not possess that luxury. For the most part, a neuron generated during your early development is the same neuron you rely on in your 80s. It is trapped with whatever metabolic waste it generates over an entire human lifespan, unless it have a highly specialized internal mechanism to clear it.
So if we think about this in practical terms, skin cells get to constantly move into brand new houses and bulldoze the old ones. A neuron is essentially a hoarder living in the exact same house for 90 years.
Yes.
It generates trash every single day just by keeping the lights on, but it can never move. It is entirely responsible for its own internal waste management.
And the architecture of that house is the second major vulnerability. Neurons have an incredibly elongated morphology.
You're weirdly shaped.
Very. They possess a central cell body, the soma, but then they have an intricate branching tree of dendrites and an axon that can stretch to astonishing lengths.
Yeah.
I mean, a single motor neuron's axon can extend from the base of your spinal cord all the way down to your toe.
Which creates a massive logistical nightmare. The literature refers to this as a cellular trafficking defect. Moving cargo from the far end of an axon back to the soma isn't just difficult. It requires an incredibly complex railway system.
It relies on microtubules and motor proteins called kinesin and dynin, and it is biologically exhausting. The disposal and recycling machinery of the cell, primarily the lysosomes, are mostly concentrated back in the central cell body.
So the trash incinerator is all the way in the kitchen, basically.
Right. So if a protein misfolds at the furthest synaptic terminal of that axon, the neuron has to physically transport that microscopic piece of trash along the microtubule track across a vast cellular distance just to break it down.
Wow.
The physical distance and the narrow, restrictive shape of the axon make moving waste highly susceptible to bottlenecks and traffic jams.
So our hoarder isn't just stuck in the same house for 90 years. They are living in an apartment that consists of an incredibly long, microscopic hallway.
Yes, exactly.
They have to drag heavy bags of garbage from the very end of the hallway all the way back to the kitchen just to put it in the incinerator.
And to compound the issue, we arrive at the third vulnerability, the sheer volume of waste generated. Neurons have a massive energetic demand.
Because they're always firing.
Right. The human brain accounts for roughly 2% of total body weight, yet it consumes 20% of the body's energy. Firing synapses, maintaining membrane potentials, and processing continuous streams of information requires astronomical amounts of ATP, the cellular energy currency.
But generating all that ATP is a messy chemical process. It produces a massive amount of metabolic exhaust in the form of reactive oxygen species, or free radicals.
Oxidative stress is the inescapable byproduct of neural function. So you have a postmitotic cell that lives for decades, structured like a narrow, elongated hallway, generating a staggering volume of toxic metabolic byproducts every single second.
It's a recipe for disaster.
It really is. When the transport machinery slows down or the incinerator in the cell body can't keep up, that garbage begins to pile up in the hallway.
And that's where the diseases start.
Yes. This failure of clearance is the fundamental underlying pathology of neurodegenerative disease. When the waste accumulates, the neuron becomes metabolically dysfunctional. Its synaptic firing misaligns. Eventually, it undergoes apoptosis and dies.
It paints a rather bleak picture of our neurobiology, honestly. But this is exactly where the concept of macrotophagy comes in. If the cell can't divide to dilute the waste, and it can't physically push the waste out of the cell, it has to rely on cellular self-digestion.
Autophagy, quite literally translating to self-eating, is the most vital survival mechanism the postmitotic cell possesses. It's the process by which the neuron identifies its own internal debris, sequesters it, breaks it down into basic amino acids and fatty acids, and reuses those components to build fresh functional structures.
But the crucial point in the research is that this system doesn't run at maximum capacity by default. Like, you don't trigger high levels of macrotophagy by maintaining a state of constant caloric surplus.
Not at all.
If you are eating 3 meals a day with snacks in between, this deeply embedded cleaning mechanism remains largely dormant, right?
Because the biological trigger for autophagy is metabolic stress, specifically nutrient deprivation. Our bodies operate on a spectrum of homeostasis. When nutrients are abundant, the body is aggressively biased toward growth, storage, and cellular proliferation.
And the primary conductor of this growth state is a protein kinase complex known as mTOR, the mechanistic target of rapamycin. Let's actually dig into how mTOR functions, because the literature doesn't just call it a switch. It's a highly sensitive nutrient sensor.
Yeah, it's tethered to the surface of the cell's lysosomes. It is constantly sampling the cytosolic environment for the presence of amino acids, particularly leucine and arginine, as well as glucose.
So it's constantly tasting the cell to see if there's food.
Pretty much. When insulin levels are high and blood glucose is elevated following a meal, mTOR is heavily activated. It signals the cell to synthesize proteins, build new structures, and suppress any breakdown of existing cellular material. It essentially tells the cell, hey, resources are infinite, keep expanding.
But when you initiate a fast, you drastically alter that chemical environment. Blood glucose drops, insulin plummets, and the amino acid concentration in the cell falls. That drop in available energy activates another enzyme called AMPK.
AMPK, AMP-activated protein kinase, is the opposing force to mTOR. It senses the drop in ATP, the cell's energy currency. When AMPK activates, it directly inhibits mTOR.
The growth signals just go completely silent.
Completely silent. The cell suddenly registers that external resources have vanished, yet it still faces the massive energetic demand required to keep you alive.
So the neuron looks down its long, narrow hallway, sees the accumulated metabolic trash, the misfolded proteins, and the old organelles, and realizes, well, I have no food coming in, so I'm going to have to burn the furniture to survive.
That's a perfect analogy. The initiation of macroautophagy is an incredibly elegant sequence of events. Once mTOR is inhibited, it triggers a cascade that leads to the formation of a structure called the phagophore.
And this isn't a permanent organelle, right?
Oh, it's transient. It's a crescent-shaped double membrane that just spontaneously forms in the cytoplasm.
The literature describes it as essentially a microscopic garbage bag. It physically expands, wrapping itself around a portion of the cytoplasm, sequestering the old, damaged cellular components.
And the mechanism by which it seals off the waste involves a complex array of autophagy-related proteins, or ATGs. Once the double membrane fully encloses the targeted debris, it seals shut, becoming a complete spherical structure called an autophagosome.
But sequestering the waste is only the first step. The autophagosome now has to travel off and along those vast microtubule railways we discussed until it meets a lysosome.
The lysosome being the actual incinerator. The lysosome is a membrane-bound organelle packed with highly acidic hydrolysis enzymes designed to dismantle complex molecules. When the autophagosome fuses with the lysosome, it becomes an autolysosome.
And that's where the magic happens.
The acidic enzymes flood the inner chamber, completely dissolving the trapped garbage. The resulting raw materials, the pristine amino acids and simple lipids, are then pumped back out into the cell's cytoplasm to be reused.
It's not just taking out the trash, it's a closed-loop recycling system. It provides the fuel required to survive the famine while simultaneously purging the neurotoxic debris that causes cognitive decline. Here's where it gets really interesting, though.
Oh, definitely.
How do we know when we've actually triggered this state? The sources highlight a specific metric used to measure this metabolic shift: the glucose ketone index, or GKI.
The GKI, developed by researcher Dr. Thomas Seyfried, offers a quantifiable window into your metabolic state. It's a simple ratio comparing your circulating blood glucose to your blood ketones.
Because when glucose runs low during a fast, the liver begins converting stored fatty acids into ketone bodies.
Right, like beta-hydroxybutyrate and acetoacetate, which easily cross the blood-brain barrier to fuel the neurons.
And the target ratio the research points to for deep therapeutic autophagy is a GKI of 1:1.
The 1-to-1 ratio, where your ketone concentration roughly equals your glucose concentration in millimolar measurements, indicates a profound state of metabolic stress. It signifies that the body has almost entirely exhausted its glycogen reserves and has aggressively shifted into a state of cellular scavenging.
The applications for this level of metabolic stress are fascinating. The literature discusses how a 1:1 GKI is highly effective at neutralizing metabolically inflexible cells, particularly tumor cells, but also senescent cells.
The Warburg effect dictates that many tumor cells rely almost exclusively on glycolysis, fermenting huge amounts of sugar to survive. They often lack the functional mitochondria required to burn ketones efficiently.
So they basically starve.
Precisely. When you plunge the body into a 1-to-1 GKI, healthy neurons effortlessly switch over to burning ketones, but the pathological cells are starved of their required glucose.
And it does the exact same thing to senescent cells. These are the cells the sources refer to as zombie cells.
Zombie cells are exactly what they are. Senescent cells are damaged cells that have permanently halted division but have evaded apoptosis. They refuse to die.
They just sit there.
They sit in the tissue, heavily reliant on glucose, continuously secreting a toxic cocktail of pro-inflammatory cytokines, known as the SASP, senescence-associated secretory phenotype. This localized inflammation accelerates the aging and degradation of all the healthy neighboring cells.
But when you induce that state of deep autophagy, the body realizes it can't afford to feed these inefficient zombie cells. It prioritizes the healthy neural networks and cannibalizes the senescent cells for parts.
If we connect this to the bigger picture, it is evolutionary triage. During famine, survival dictates extreme efficiency. Macroautophagy is the system prioritizing the collective health of the organism by dismantling its weakest, most toxic components.
So macroautophagy acts as the generalized, citywide waste management system. It's handling the bulk clearance, the cytosolic debris, and the recycling of zombie cells. But as we transition deeper into the pathology of neurodegeneration, the sources reveal that the brain faces highly specialized threats that a standard autophagosome cannot handle alone.
Right, you need special tools.
Exactly. This brings us to the targeted, selective forms of autophagy, mitophagy and xenophagy.
While macroautophagy can sweep up bulk cytoplasm, the cell requires highly selective mechanisms to deal with complex, localized disasters. Mitophagy is the targeted identification and destruction of failing mitochondria.
We established earlier that the energetic demand of the neuron is staggering, meaning the mitochondria are under immense functional strain. They are burning glucose and ketones, generating ATP, but inevitably leaking reactive oxygen species.
And those free radicals aggressively damage the mitochondrial DNA and the structural proteins of the organelle itself. Over time, the mitochondrion begins to fail.
It starts falling apart.
Yes, it loses its membrane potential, its ability to generate ATP plummets, and its emission of toxic free radicals skyrockets. It essentially becomes a localized chemical fire inside the neuron.
And if we look at the clinical literature, mitochondrial dysfunction isn't just a minor issue. It is heavily implicated in a terrifying array of neurological conditions. Parkinson's, Alzheimer's, Huntington's, ALS, even chronic fatigue syndrome all showcase severe mitochondrial degradation.
If the power grid within the neuron collapses, the neuron cannot maintain its synaptic connections, and the network begins to unravel. Mitophagy is the emergency response. The neuron utilizes a specific biochemical pathway, often involving proteins like PNK1 and parkin, to detect when a mitochondrion has lost its membrane potential.
How does that actually work chemically? Like, how does the cell know a specific mitochondrion is failing?
Under healthy conditions, the PNK1 protein is constantly imported into the mitochondria and degraded.
Right.
But when the mitochondrion is damaged and loses its voltage gradient, PNK1 accumulation occurs on the outer membrane.
Oh, so it acts like a distress flare.
It acts exactly like a distress flare. It recruits the parkin protein, which then attaches ubiquitin molecules to the surface of the broken organelle.
And ubiquitin acts as a molecular tag that essentially screams, "Destroy me."
Exactly. The autophagic machinery recognizes the ubiquitin tag. A specialized phagophore forms directly around the failing mitochondrion, isolating it from the rest of the cell, and delivers it to the lysosome for total degradation.
That is incredible.
Once the damaged unit is cleared and broken down into its base amino acids, the neuron can undergo mitochondrial biogenesis, synthesizing a brand new, highly efficient power plant to replace it.
If macroautophagy is the routine garbage collection, mitophagy is the hazardous materials team specifically trained to dismantle a leaking nuclear reactor before it melts down the entire facility.
A very precise analogy. And we carry that forward, xenophagy operates as the internal cellular immune response. It is the targeted clearance of intracellular pathogens, viruses, and bacteria that have successfully breached the outer membrane of the cell.
This is a massive paradigm shift for how most of us understand the immune system. I mean, we usually picture white blood cells and antibodies floating around in the bloodstream, fighting infections in the extracellular space. But viruses don't stay in the bloodstream, they hijack the inside of the cell.
Viruses are obligate intracellular parasites. They must enter the host cell to utilize its replication machinery. While circulating antibodies can neutralize free viral particles, they cannot penetrate a living human cell to extract a virus that has already set up shop inside.
The sources bring up the varicella zoster virus as the prime example. Most people encounter it as chickenpox in childhood. The immune system mounts a defense, the spots disappear, and you think you're cured.
But you are not. The VZV virus retreats and establishes latency within the sensory ganglia, specifically in the nerve cells of your dorsal root or cranial nerves.
It just hides.
It inserts its genetic material and lies completely dormant, shielded from circulating antibodies, for decades.
It's hiding out in the hoarder's hallway.
Yes. And years later, usually precipitated by acute stress, aging, or a temporary dip in immune function, the virus reactivates. It travels back down the axon to the skin and causes the excruciatingly painful condition we know as shingles.
But the research indicates that fasting-induced xenophagy gives the neuron a mechanism to actively hunt down these latent intruders.
When nutrient deprivation triggers high-level autophagy, the cell begins aggressively scanning its internal environment for anything it can break down for raw materials. The autophagic machinery can actually recognize foreign viral capsids or bacterial components floating in the cytoplasm.
Wow, so it forms a phagophore around the pathogen.
Delivers it to the lysosome, and uses the acidic hydrolases to literally digest the virus. It reduces the intracellular viral load by converting the invader into fuel.
That biological mechanism completely explains the anecdotal experience of being severely ill. Think about the onset of a brutal flu or a severe gastrointestinal virus. The very first systemic symptom is profound anorexia. You completely lose your appetite. The mere smell of food induces nausea.
The clinical term is the anorexia of infection. It is an evolutionary, highly conserved, acute phase response.
Your body is intentionally enforcing a fast. It's hijacking your behavior to instantly drop blood glucose and shut down mTOR. It knows that cutting off the nutrient supply forces the cells to turn inward, activate xenophagy, and aggressively digest the viral particles to survive.
It's not a side effect of the illness. It is the primary biological protocol for curing it.
That is a staggering realization.
It really reframes how we look at being sick.
It does. So we have mechanisms to clear the general debris and the zombie cells, mechanisms to swap out the failing mitochondrial power plants, and mechanisms to digest invading viruses. But there is one final, specialized clearance system the sources focus on, and it directly addresses the most terrifying hallmark of neurodegenerative decline, the slow structural accumulation of misfolded proteins. We need to examine agrophagy.
Agrophagy is arguably the most critical autophagic process for preserving long-term cognitive function. It is the selective clearance of aggregate-prone proteins.
Let's break down the chemistry of an aggregate. Why do proteins misfold in the first place, and why do they clump together?
Well, proteins are long chains of amino acids that must fold into highly specific three-dimensional shapes to function properly. That folding is dictated by their chemical sequence and assisted by cellular chaperones.
Okay.
But under conditions of high oxidative stress, aging, or genetic mutation, proteins can misfold. Instead of their proper functional shape, they often expose sticky hydrophobic regions that should be hidden inside the molecule.
And because they're hydrophobic, they desperately want to stick to other similar proteins to avoid the watery environment of the cytoplasm. They form what are called beta sheet structures.
Exactly. They begin to stack on top of one another, forming oligomers and eventually massive insoluble fibrils and aggregates. These clumps act like biological concrete pouring into the narrow hallways of the neuron's axon.
Oh man, biological concrete.
It's devastating. They disrupt intracellular transport, trap other vital proteins, and eventually trigger apoptosis.
The sources list the specific proteins notorious for this behavior, and it reads like a catastrophic roll call. Mutant HTT proteins responsible for Huntington's disease, SNCA or alpha-synuclein, the primary component of Lewy bodies and Parkinson's disease. And of course, MAPT, better known as tau tangles, alongside amyloid beta plaques, which define Alzheimer's disease.
These aggregate-prone proteins are the physical manifestations of the diseases. Without robust agrophagy, the clumps grow exponentially. The research highlights incredibly compelling data from animal models demonstrating how autophagic failure accelerates this process and how inducing autophagy can reverse it.
Let's look at the Lurcher mice study mentioned in the literature.
Yeah, the Lurcher mutation in mice affects specific glutamate receptors, leading to chronic excitotoxicity and a severe impairment of autophagy, specifically in Purkinje cells, large neurons in the cerebellum critical for motor control.
Right, so because autophagy is impaired, the Purkinje cells rapidly accumulate aggregates, become completely dysfunctional, and die en masse, leading to severe ataxia in the mice.
It provides direct proof that without the autophagic clearance system, the neuron is doomed. But the researchers didn't stop there. They looked at models of Huntington's disease, utilizing both transgenic mice and Drosophila fruit flies.
Huntington's is caused by a massive expansion of a polyglutamine tract in the Huntington protein, making it highly prone to aggregation.
Right. So the researchers administered rapamycin to these animal models.
And rapamycin is the compound that directly inhibits mTOR. It artificially mimics the exact cellular state of a prolonged fast without altering the animal's diet.
Yes. By chemically suppressing mTOR, they forced the neurons into a state of rampant agrophagy. The autophagosomes began aggressively targeting and engulfing the mutant Huntington aggregates.
The result was a significant reduction in the toxicity of these polyglutamine expansions and a marked improvement in the neurodegenerative symptoms of the animals. They utilized the cell's starvation response to eat the very structures causing the disease.
But this raises an important question. What happens if the garbage disposal itself breaks?
The Alzheimer's research points out a terrifying potential bottleneck in this entire system. You can bag up the trash all you want, but if the incinerator is broken, the whole system collapses.
This is a critical nuance in the pathology of Alzheimer's. We mentioned earlier that the lysosome requires a highly acidic environment to digest the waste. That acidity is maintained by specific proton pumps called VAT paces on the lysosomal membrane.
Right, and the research indicates that in certain models of Alzheimer's, a primary failure occurs at those pumps. The lysosomal acidification process fails.
The pH inside the lysosome rises, rendering the digestive hydrolases useless. So the FIGA4 successfully recognizes the misfolded amyloid beta, it engulfs it, and it fuses with the lysosome to create the autolysosome. But because the environment isn't acidic, the amyloid beta isn't destroyed.
It just sits there. The cellular garbage bags just start piling up inside the neuron, completely undigested.
It creates a massive catastrophic bottleneck. The neuron becomes physically engorged with these massive failed autolysosomes, a condition known as neuritic dystrophy.
It's like a balloon filling up with trash.
Exactly. And it is exactly this localized cellular failure that eventually bursts the neuron, spilling the undigested amyloid beta into the extracellular space, which yields the massive senile plaques characteristic of late-stage Alzheimer's.
The implication there is profound. If you maintain regular periods of metabolic stress, if you practice intermittent or prolonged fasting throughout your life, you are continuously exercising that autophagic flux.
You are.
You are keeping the VAT pace pumps active, ensuring the lysosomes remain acidic, and clearing the amyloid beta before it ever has a chance to form those catastrophic bottlenecks.
You are maintaining the structural integrity of the clearance pathway. But it's also vital to understand that the neurons are not facing this toxic burden in isolation. The brain operates as a highly integrated community.
Which brings us to the fascinating support network explored in the sources. The brain isn't just a web of neurons. It relies on an entire neurogenitorial and structural support staff, collectively known as the glia.
Glial cells are the non-neuronal cells of the central nervous system. For decades, neuroscience viewed them as simple connective tissue, the glue holding the brain together. We now know they are active, dynamic participants in neural health, and crucially, they are just as dependent on fasting-induced autophagy as the neurons themselves.
The sources categorize three main players here. Let's start with the microglia.
Microglia are the resident macrophages, the primary immune cells of the brain. While neurons utilize agrephagy to clean their own internal cytoplasm, microglia utilize autophagy to fuel their external scavenging.
They are highly mobile, extending and retracting processes to constantly survey the brain tissue, right?
Yes. And when a neuron dies and leaves behind that senile amyloid plaque we just talked about, it's the microglia's job to clean up the mess in the extracellular space.
They physically phagocytose. They engulf and eat the extracellular debris, dead cells, and misfolded protein plaques.
But to digest what they eat, their internal autophagic machinery must be highly functional. If microglial autophagy is defective, they ingest the amyloid but cannot break it down. They become overwhelmed, senescent, and flip from being protective scavengers to chronic sources of neuroinflammation.
Then we have the astrocytes, which are heavily involved in the metabolic coupling of the brain.
Astrocytes have processes that wrap around both the neuronal synapses and the brain's capillaries. They literally pull glucose from the bloodstream, process it into a more efficient fuel called lactate, and hand it directly to the neurons.
But the research highlights that when autophagy is suppressed in astrocytes, they undergo severe metabolic failure.
They cease their supportive functions and transition into a reactive, pro-inflammatory state. They stop feeding the neurons and instead release toxic molecules that can accelerate synaptic degradation.
Finally, the sources look at the oligodendrocytes. These are the cells responsible for myelination.
Myelin is the lipid-rich, fatty sheath that wraps around the axons of the neurons. It acts as electrical insulation. Without myelin, the electrical action potentials would dissipate and signal transmission would be impossibly slow.
It's the rubber coating on the copper wire. If it degrades, the signal shorts out, which is the underlying cause of demyelinating diseases like multiple sclerosis.
And the research is explicit. Robust autophagic flux within the oligodendrocytes is absolutely essential for proper myelin compaction and maturation. Without autophagy clearing out the cellular workspace, the oligodendrocytes cannot produce the complex lipid layers required to insulate the brain.
Wait, I need to pause here and look at the actual genetic mechanisms the literature mentions because this ties the whole glial network together. The sources cite studies involving the genetic manipulation of core autophagic genes, specifically BECN1, ATG5, and ATG7.
Yes. BECN1, or Beclin-1, is a critical protein involved in the very first stages of forming the phagophore membrane. ATG5 and ATG7 are part of the complex conjugation system that allows that membrane to physically elongate and seal shut.
So if you genetically upregulate those specific genes in animal models, you are artificially installing a more aggressive, high-capacity garbage disposal system across all these cell types.
Exactly. And the literature shows that when those genes are upregulated, which is exactly what the systemic drop in insulin inactivation of AMPK does during a faster, the entire glial network optimizes.
Microglia clear plaques faster, astrocytes improve their metabolic support, and oligodendrocytes maintain thicker, healthier myelin sheaths.
The fasting state orchestrates the health of the entire neurological ecosystem.
It is a stunningly cohesive biological narrative. We have mapped out the clearance mechanisms. Macroautophagy sweeps the cytosol, mitophagy replaces the degraded mitochondria, xenophagy hunts the latent viruses, and agriphagy dissolves the toxic protein clumps, all while the glial cells scrub the extracellular space.
It's quite the operation.
We have meticulously detailed the demolition and waste removal, but demolition alone doesn't restore a crumbling building. To truly heal, the brain must rebuild. And this brings us to the final and perhaps most miraculous phase of this biological sequence, neurogenesis and the role of BDNF.
For a significant portion of modern medical history, the prevailing dogma was that adult neurogenesis, the creation of brand new neurons, was impossible.
Really?
Yeah, the scientific consensus held that you were born with a finite allocation of brain cells, and adulthood was simply a slow, managed decline as those cells inevitably died off.
Well, we now know that is fundamentally incorrect. The adult brain possesses distinct neurogenic niches, specifically in the dentate gyrus of the hippocampus, the area governing memory and learning where neural stem cells reside, capable of differentiating into fully functional brand new neurons.
But those stem cells remain largely quiescent unless they receive the proper biological signal to activate. And the literature repeatedly points to metabolic stress, specifically fasting, as one of the most potent triggers for that activation.
The sources draw a direct physiological parallel between fasting and exercise. They operate on the exact same principle of hormesis.
Hormesis is the biological phenomenon where a low to moderate dose of a stressor provokes an adaptive, highly beneficial response in the organism. If you want to increase muscle mass, you have to subject the muscle fibers to the physical stress of resistance training, creating microtears.
And the body responds to that stress by overcompensating, building denser, stronger tissue.
Fasting is the cognitive equivalent of lifting weights. You subject the neural network to the metabolic stress of acute energy deprivation.
The brain registers the stress, triggers the autophagic clearance we discussed, but then follows it up by initiating an aggressive growth protocol to ensure the network is stronger and more resilient for the next famine.
The animal models demonstrating this are remarkable. The sources reference a study published in the Journal of Cerebral Blood Flow and Metabolism involving mice placed on an intermittent fasting regimen for 3 months.
They tracked 3 distinct metrics: cell production, cell death, and overall neurogenesis. The results showed a massive, quantifiable increase in the generation of new neural tissue in the fasted mice compared to the ad libitum, constantly fed control group.
More importantly, the newly generated network was structurally superior. When those fasted mice were later subjected to induced strokes, they suffered significantly less infarct volume, less brain cell death than the control mice. The stress of the fast had forged a deeply resilient neural architecture.
And the chemical architect coordinating all this new construction is a protein called BDNF, brain-derived neurotrophic factor.
BDNF is arguably the most vital molecule for brain health. It is a neurotrophin, essentially acting as a highly potent fertilizer for the central nervous system.
It binds to specific receptors, TRKB receptors on the surface of neurons, initiating intracellular cascades that prevent apoptosis, promote the growth of new dendrites, and facilitate long-term potentiation.
Which is the physical basis of learning and memory consolidation.
And its impact on mood is profound. The sources highlight that BDNF acts as a powerful natural antidepressant, which makes evolutionary sense. If you are starving, your brain floods with BDNF to increase your cognitive focus, spatial memory, and motivation to go hunt and find food.
You can't afford to be lethargic when resources are scarce. The data on fasting and BDNF expression is striking. In the murine models we just discussed, fasting protocols increased BDNF levels in the brain by up to 50%.
That is a massive systemic flood of neurotrophic growth factor triggered simply by the absence of calories, and this translates directly to human clinical outcomes. Let's look at the Alzheimer's study detailed in the sources.
Right, the researchers took 10 human subjects who were already exhibiting documented cognitive decline, early signs of Alzheimer's.
These were individuals already experiencing the manifestation of the aggregate accumulation and autophagic failure we outlined earlier. The researchers instituted several lifestyle modifications, but the foundational protocol was implementing a strict 12-hour nightly fast.
Which is a very manageable intervention.
Wait, I need to push back on the biology here because earlier we established that to achieve a GKI of 1 to 1 and trigger deep, rampant autophagy, you generally need to fast for days, completely depleting liver glycogen. A 12-hour fast from dinner to breakfast won't get you anywhere near a 1 to 1 GKI. So how is a mere 12 hours reversing cognitive decline?
It is an excellent point of physiological distinction. A 12-hour fast does not induce deep systemic macroautophagy. However, it does accomplish two critical things. First, it halts the continuous activation of MPR.
So it gives the body a break.
It gives the body a daily window where insulin levels drop to baseline, allowing the cellular machinery to begin basic maintenance, even if it doesn't reach deep scavenging mode. Secondly, and perhaps more importantly for early cognitive decline, it tightly aligns food intake with the circadian rhythm.
By stopping the intake of food 3 hours before sleep, you allow blood glucose to stabilize.
Exactly, which in turn allows the brain to optimize its lymphatic system, the fluid clearance system that flushes out the extracellular space during deep sleep. Even mild periods of insulin lowering and mild metabolic stress begin the cascade of BDNF production.
And the results of that study speak for themselves. After just 6 months of adhering to that 12-hour fasting window, 9 out of the 10 subjects demonstrated quantified, documented improvements in their cognitive abilities. They successfully reversed the trajectory of their decline.
A 90% success rate in improving cognitive function for a disease that currently possesses zero effective pharmaceutical cures.
It's astounding. But the sources do acknowledge a caveat. While a 12-hour or 16-hour daily fasting window is an excellent baseline for maintenance and mild neurogenesis, it may not be a powerful enough intervention for someone facing severe entrenched metabolic dysfunction or advanced neurodegeneration.
If the autolysosome bottleneck is already severe or the senescent cell burden is excessively high, mild intermittent fasting won't generate the intense metabolic stress required to trigger massive cellular apoptosis or deep agrophagy.
So to achieve that therapeutic level, you generally must engage in prolonged fasting 48 to 72 hours or longer of only consuming water.
But prolonged water fasting presents significant hurdles. It is incredibly demanding psychologically and physiologically.
And for older demographics or individuals with pre-existing comorbidities, the potential for dangerous electrolyte imbalances, severe hypoglycemia, or excessive lean muscle loss makes prolonged water fasting clinically risky.
Which is why the sources dedicate significant attention to a vital scientific intervention, the fasting mimicking diet, or FMD.
The FMD was developed through extensive research by Dr. Valter Longo at the University of Southern California with significant funding and backing from the National Institutes of Health. It is an incredibly elegant solution to the dangers of prolonged water fasting.
Dr. Longo's research team spent years reverse engineering the body's nutrient sensing pathways. The FMD is a highly specific, mathematically formulated plant-based dietary protocol lasting 5 days.
The brilliance of the FMD lies in its macronutrient ratios. It provides the patient with roughly 700 to 1,100 calories a day, enough food to prevent dangerous muscle catabolism and provide essential micronutrients.
But because the diet is extremely low in the specific amino acids that trigger mTOR and strictly regulates the carbohydrates that spike insulin, the cellular sensors remain entirely blind to the calories.
The patient is eating physical food, but on a biochemical level, the cells register a state of absolute starvation.
The AMPK pathway activates, mTOR remains inhibited, and the body descends into deep macroautophagy, mitophagy, and agrophagy, exactly as it would on a pure water fast, but with a vastly superior safety profile and much higher patient compliance.
The clinical trials outlined in the sources demonstrate massive systemic benefits. Significant reductions in visceral fat without the loss of lean muscle mass, marked drops in blood pressure, major improvements in systemic insulin resistance, and a dramatic lowering of IGF-1, an insulin-like growth factor heavily implicated in aging and oncogenesis.
But the most crucial element of the FMD protocol, and of any prolonged fast, isn't just the deprivation phase.
No, the most profound biological magic occurs the moment the fast is broken. The refeeding phase.
The literature stresses that the true magnitude of neurogenesis and cellular renewal isn't realized while you are starving. The fast is purely the demolition phase.
It forces the cell to break down the misfolded proteins, the viral capsids, and the broken mitochondria, leaving behind a massive pool of highly purified raw materials, the scavenged amino acids and lipids.
But the body cannot build the new structures until it receives the signal that the famine has ended. When you reintroduce nutrient-dense food after a prolonged fast, or the FMD, insulin and blood glucose rise and mTOR is violently reactivated.
But now it's activating in a cellular environment that has been completely scrubbed clean of senescent cells and toxic debris.
The sudden influx of external nutrients combined with a massive pool of recycled internal materials triggers the activation of dormant stem cells.
The BDNF that spiked during the fast orchestrates the use of all those resources to rapidly construct brand new, highly efficient mitochondria, pristine functional proteins, and entirely new neural connections.
It is the ultimate biological upgrade. You subjected the system to the crucible of metabolic stress, forcing it to tear down the rotting, dysfunctional architecture. Then, through the precise reintroduction of nutrients, you instructed the stem cells to construct a resilient, upgraded network in its place.
The cycle of demolition, followed immediately by robust reconstruction, is the fundamental biological engine of longevity and cognitive preservation.
And it requires both phases to function. Chronic overfeeding prevents the demolition. Chronic starvation prevents the reconstruction. The healing intelligence of the brain relies entirely on the oscillation between the two states.
So what does this all mean? We have completely mapped the territory. We started with the vulnerable postmitotic neuron, trapped in its elongated morphology, suffocating under the weight of its own metabolic exhaust.
And we watched how the simple absence of nutrients activates a cascading symphony of survival.
Macrotophagy bags up the cytosolic debris. Mitophagy systematically replaces the failing power plants. Xenophagy actively digests the viral invaders hiding in the network. Agraphagy dissolves the sticky neurotoxic plaques before they can shatter the cell.
The glial network scrubs the extracellular space and fortifies the myelin insulation.
And finally, BDNF and stem cell activation utilize the scavenged wreckage to build a vastly superior, deeply resilient neural architecture.
It is an awe-inspiring demonstration of evolutionary engineering.
The takeaway from all this dense biological mechanics is incredibly empowering. You possess a built-in, fully integrated cellular repair facility. You are not a passive victim to the accumulation of neurological damage.
The power to clear the brain fog, to actively protect your synaptic pathways, and to physically regenerate your cognitive hardware is quite literally encoded into your DNA right now. It possesses the full blueprint for its own restoration. It simply requires you to provide the correct metabolic signal to initiate the protocol.
Which brings us to a profound, almost uncomfortable realization about how we live today.
If human biology, if the intricate architecture of our neurology, evolved to absolute dependence on the acute metabolic stress of fasting and famine, to activate its most vital repair, clearance, and growth mechanisms, we have to look critically at our modern environment. Are the comforts of industrialized society, our culture of perpetual 247 snacking, and our unyielding absolute abundance of readily available calories actually the most unnatural and dangerous environment we could possibly expose our brains to?
It fundamentally flips the script on what we consider healthy. We have systematically engineered away the exact evolutionary stress required to keep our minds from decaying. Think about the biology the next time you find yourself reaching into the pantry late at night just out of habit. Your brain is not a static machine destined to simply wear out. It is a highly dynamic, living ecosystem begging for the metabolic space to clean its own waters. Thank you for joining us on this deep dive.